For a long time, human intuition suggested that biological inheritance was a blending process, like mixing two colors of paint. But the reality is far more rigid. Traits are passed down through a hidden system of distinct, indivisible information. We know this thanks to an Austrian monk named Gregor Mendel. In the mid-19th century, he initiated a series of hybridization experiments that mapped exactly how this information travels between generations. He started by isolating true breeding variations of pea plants. These were plants that reliably produced identical offspring over multiple generations. He focused on contrasting easily measurable physical traits, specifically pitting true breeding tall stem plants against dwarf stem plants. Mendel crossed the tall and dwarf parents to create the first filial or F1 generation. The result was absolute. Every single plant in the F1 progeny grew tall. The dwarf trait did not dilute the height of the offspring to create a medium-sized plant. It vanished completely from visual observation, defying the assumption that traits inevitably blend. To see if the dwarf trait was actually gone, Mendel took the next logical step. He allowed those uniformly tall F1 plants to self-pollinate. This generated the second filial, or F2 generation, and produced a surprising outcome. The vanished dwarf trait reappeared, sorting the offspring into a precise mathematical ratio. 3/4 of the plants were tall, and exactly 1/4 were dwarf. These F2 dwarf plants exhibited no blending. Their physical height was a perfect identical match to the original dwarf grandparent from the initial cross. This proved that biological traits are governed by stable, discrete units of information. They are passed down entirely unchanged, capable of remaining dormant in one generation, only to express perfectly in the next. Mendel referred to these discrete units as factors. Modern biology calls them genes. And because a single gene can hold contrasting information, like tallness versus dwarfness, we call those different versions alleles. This distinction requires separating an organism into two layers. The physical, visible expression of a trait is its phenotype. The underlying, invisible genetic code that produces it is its genotype. We map these factors using standard alphabetical variables. A capital T represents the tall allele, and a lowercase t represents the dwarf allele. Because plants inherit one allele from each parent, they form pairs, TT, TT, or TT. This brings us to the law of dominance. When a dominant allele, capital T, is paired with a recessive allele, lowercase t, the dominant allele completely masks the physical expression of the recessive one. The plant grows tall. Because of dominance, physical appearance is an unreliable narrator. Two plants can share an identical tall phenotype, yet harbor entirely different hidden genotypes. To understand how these alleles move from parent to offspring, we have to shift our perspective away from the macroscopic plant down into the microscopic mechanics of cellular reproduction. During the process of meiosis, parent plants produce sex cells, or gametes. The law of segregation states that during this division, paired alleles split apart. Each resulting gamete receives only one allele from the pair. This segregation is an entirely random mechanism. It operates strictly like a coin flip. This graphic shows the probability map for a heterozygous plant, one carrying a capital T and a lowercase t. Every time it produces a gamete, there is exactly a 50% chance it will carry the dominant T allele and a 50% chance it will carry the recessive T allele. The biological division of cells ties directly to mathematical constraints. Reproduction acts as a highly predictable probability engine. We can calculate the overlapping probabilities of these separating gametes using a graphical tool called the Punnett square, developed by British geneticist Reginald Punnett. To model the self-pollination of the heterozygous F1 clans, we set up a 2x2 matrix. We place the 50-50 female gamete probabilities on one axis and the male probabilities on the other. Filling in the intersecting squares reveals the hidden genotypic ratio created by random fertilization. We get 1/4 capital TT, 1/2 heterozygous TT, and 1/4 lowercase tt, a strict 1:2:1 genotypic ratio. When we map these genotypic outputs back to their physical phenotypes, the math explains the macroscopic reality. Because T dominates T, 3 of the 4 squares produce a tall plant, leaving exactly 1 square to produce a dwarf plant. This is the origin of the 3:1 ratio. This Punnett matrix translates directly into pure algebra. The intersections are simply the expansion of a binomial expression. 1/2 t plus 1/2 t squared. Expanding that equation yields the exact same genotypic fractions. Mendelian inheritance is a robust mathematical system where the physical traits of a living organism align perfectly with the laws of binomial probability. The law of dominance creates an inherent blind spot for researchers. Visual observation alone cannot confirm an organism's true genetic makeup. This diagram illustrates the problem using flower color. If you are observing a dominant phenotype, like a violet flower, how can you definitively prove whether it holds a homozygous double capital V genotype or a heterozygous capital V lowercase v genotype? Merely self-pollinating the plant and waiting generations to see if a recessive white flower pops up is inefficient. In smaller sample sizes, it leaves far too much room for statistical error. Biologists require a faster, predictive experimental design, one specifically constructed to force any hidden recessive alleles out of hiding immediately. The solution is a methodology known as the test cross. In this experiment, the organism showing the unknown dominant phenotype is bred strictly with a homozygous recessive parent. The logic of this design relies on the recessive parent acting as a genetic blank canvas. It only provides recessive alleles to the offspring, which are entirely incapable of masking the unknown parent's genetic contribution. If the unknown plant is homozygous dominant, it will only pass on dominant alleles. When mapped on a Punnett square, we see this ensures 100% of the offspring will exhibit the dominant violet phenotype. In the alternative scenario, if the unknown plant is heterozygous, the results diverge. The test cross produces exactly a 50% violet and 50% white phenotypic split in the offspring. That white phenotype definitively proves the original parent was hiding a recessive allele. Through the laws of dominance and segregation, Mendelian genetics turns biological inheritance into a solvable equation. It establishes a framework that allows us to predict the future outcomes of reproduction and reverse engineer the hidden genetic past of living organisms.